Team Taser approved!
Justin Smea's Bigfoot DNA sample was tested by two different, there's the Camp Ketchum, and then there was the Trent University, and Camp Ketchum says it's Bigfoot, with mitochondrial being human, and blah, 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 and then there's Trent saying, bear.
Right.
Okay, Judge Judy, you're on. What is it?
Well, this was the issue that reared its head very early on when Derek invited me down to the quote-unquote kill site, the alleged kill site, because when Justin produced this little Ziploc bag that had a postage stamp size piece of the hide and hair and handed it to me, I put a 15-power loop on it and took a look, and I looked up and looked at Justin and I said, well, this isn't primate hair. And he immediately backpedaled and said, well, I never said it was Sasquatch hair. This is just what I dug up, when he came back to the scene at the urging of Derek Randalls and brought his dog, his bear hunting dog, presumably. And wherever the dog showed interest, he dug in through the 2.5 feet of snow and came up with this piece of hide. It wasn't a steak. I don't know why it keeps getting characterized as a steak. If there was any muscle tissue, it was very limited. It was mostly just skin and adipose and hair attached. Well, and then I came back to the lab and put it under the scope, and there was clearly differentiation between very coarse guard hairs.
That's what I mentioned.
50% wider than human hair, and Sasquatch, well, what we think is Sasquatch, and qualify that. What we think is Sasquatch is very similar in its coarse, or its fineness. It's about 65 microns across in diameter. And this was, 90 to 100, and long, tapering guard hairs, and very fine, very narrow underfur that was, only about 20 microns across, that had a serial stepped ladder medulla, and absolutely devoid of any pigment in the underfur. So it was white and translucent as could be.
Dr. Meldrum?
Yeah.
That brings up a point. I'm sorry to interrupt, but you were talking earlier about, the altering, I guess you call it, of samples from extensive testing. Do you think that is how the Ketchum study got the Sasquatch results that they desired on that same sample?
I honestly have no idea how they got their results. I mean, there have been all sorts of claims about the work done in these laboratories and so forth. I think their revealing discussion was one that I posted a link to on my Facebook page, where these PhD students and one PhD who was the principal voice of the group as they were ruminating over this whole thing, he pointed out that, and clearly he had some experience and knew what he was talking about dealing with these types of samples, but he pointed out that everything pointed to this just being a mishmash. That when he did a blast search of GenBank using what data he had, and he had to re-enter all her data because he pointed out that it was provided as a PDF file instead of some other type of a file that you could read the data directly from. He had to literally re-enter it. But anyway, in doing the blast, it came up with all kinds of things, including panda bear. And people wonder, how in the world would there be panda bear? Well, he very astutely pointed out that panda bear is the only bear representative for which the entire genome has been sequenced and submitted. There have been a few isolated studies of particular loci, particular genes from the bear genome that have been sequenced and have been uploaded, but the blast then found similarities between parts of her samples, of her sequence rather, and panda, and that just means that there was bear in there, and that the bear showed similarities to panda. which is closely related. So, that I'm quite confident that was almost certainly Justin's sample.
Mhm.
So these stories that there have been different samples, that Justin sent something to Melba and something to me and something to Tyler and Bart, I can't comment on. I have no way of knowing one way or the other. But the point is that if everything is above board, and if the sample that Justin recovered is in fact from Melba's description, it sounds like it's one of the 3 genomic sequences, or it was used in producing one of the 3 genomic sequences. The fact that it was quote unquote consistent with all the others, and that's an interesting point too. I mean, she cites 109 samples that she received, and all of them were consistent. I mean, when I asked for hair samples to be sent to Brian Sykes for his project, I got a lot of people sent them directly to him, but I got maybe 9 samples, and of those, immediately, I think it was 5 of the 9 were eliminated as just other common wildlife. So, and that's my experience, that when people pick up a sample in the field, the chances of it being a Sasquatch, even if there was a credible encounter or a footprint find or some circumstantial evidence or circumstances to suggest the Sasquatch was in the vicinity, the chances of it being a Sasquatch hair sample are pretty slim. I mean, there's hair all over in the environment out there. It's ubiquitous. I like to point out there's a book on tracking, this one by Resendez, and I'm sorry, maybe it's Elrod. He makes the point that he would challenge his students when they had taken a break for lunch, sat down, before they could get up, they had to find 10 strands of hair. And, that seems like a ridiculously impossible challenge, but he said once you get a search image for the hair, he said it's all over the place. It persists in the environment for a long time. That hair might not be very good for DNA analysis, and that can cause a problem. Strands of hair are picked up, and then the DNA is degraded through exposure to the sun and elements and so forth. So back to the point at hand, it was just simply the fact that she claimed that she gave no disclaimer about discounting a significant fraction of the samples that were submitted to her. That in itself is remarkably suspicious. I just find that hard to believe that every toenail, every scrap of bone, every bit of hair, every bit of blood that she received turned out to be Sasquatch. So I tell you that the Bigfoot community has gotten very efficient in collecting forensic evidence then.
Okay, well, I have an additional question kind of that trails off of that. This question is from David Bettdorf, and it kind of goes hand in hand with the Ketchum study and the claims being made by David Paulides at his lectures, or he's speaking all over the country. Of all the weirder things that people claim to experience, like eye shine, eye glow, bioluminescence, mind speak, infrasound, zapping, cloaking, etc., which, if any, do you think has a sound basis in science, and why?
Well, gee, of those you've enumerated, I'd have to say that the one that jumped out was the potential for infrasound. I still think that's a really plausible behavior for a large ape. This is an area of study, and again, it's on my kind of bucket list of things to address, because, and I touch on it in my book, because there's only very limited literature, limited research that's been conducted, and that is the presence of these extralaryngeal air sacs in the great apes. And they're quite extensive. I mean, you go back to the classic anatomical literature of the apes in anthropological journals and whatnot. There have been some studies to look at the extent of these, and they're quite extensive in gibbons and siamongs and orangutans. They're present to a lesser degree in gorillas and only rarely in chimpanzees. And in humans, they are almost never present, but they are anomalously every now and again. And then if you watched, what was that movie now? I just went blank. Oh, shucks. The one with Hurt. Um, he comes out of his isolation tank, having externalized his experience as a hominid, and can't talk. And he has they X-ray his neck, and he has laryngeal air sacs. And that was one of my first exposures to this whole concept that primates have these extra air sacs. But if Sasquatch is a close relative of the apes and or humans, then it would conceivably have such extralaryngeal air sacs. Now, and the degree of development would vary depending on the position within the phylogeny and its adaptations and selection for the use of those air sacs. Now, the air sacs can serve as resonating chambers. It can be used in they can be used in producing loud calls by sustaining the air volume and the exhalation. If they're infralaryngeal, then all the air in those sacs, as it's expelled, will pass across the vocal tract to the vocal cords.
So, do you think, Doctor, that with these infrasound capabilities, combined with a person's anxiety and panic attack, it just adds to their fear, their paralyzing fear?
That's exactly right. Because I often wondered when you would talk to these individuals, and some were experienced outdoorsmen, they were experienced hunters, they quote weren't afraid of the dark type of guys and women, and yet they would describe this otherwise inexplicable fear, this visceral response to their experience, and they swear they'll never go back there again, or they never go in the woods without a firearm there. I'm wondering what could they have experienced that would have such a traumatizing effect, and the physiological and psychological effects of exposure to infrasound could explain that. Now, the less plausible aspects of it is that usually it requires very high decibels of that vocalization to have those kinds of effects. And so the question is, could they be exposed to that intense of a blast? And there certainly are examples of animals, predators, like tigers, that use a blast of infrasound to partially incapacitate or stun their prey as they're making their leap, their final approach on the prey item. So that's it certainly is possible. Definitely is possible.
We've got like 100 questions to ask you from a lot of our friends and Team Taser members. One is from Bobby Keene. She says that where'd she go? There she is. She says hello and wants to know, where do you stand in Bigfooting? As an expert or a scientist that wants to know more, where do you place yourself in this field?
Well, I certainly wouldn't give myself the label of expert. I don't know.
Well, we give you that label.
It's a subject that has a lot of unanswered questions, and so certainly I want to know more. I mean, I do. And partly for that reason, I've always kind of remained aloof from the various organizations. It's not that I'm antisocial. It's just that I don't want my posture and position and reputation to be used by other groups, to lend credibility to their efforts when I'm not even directly involved with them. And so I'm always make myself available as a resource to such groups and individuals. And but my feet are squarely in the scientific community, and addressing this as a question of anthropology and primatology. And as I said, it dovetails so elegantly with my pre-existing research program, which was primate functional morphology in the broadest sense, and the evolution of homobipedalism specifically. I mean, here's the possibility of the existence of another biped, which could have very well evolved bipedalism completely independently. But even if it hasn't, I mean, we're beginning to recognize as anthropologists that bipedalism was not just a one-size-fits-all, that the adaptations of various and diverse hominids may have employed different strategies and different morphologies to accomplish their specific objectives and adaptations. So either way, it's a fascinating independent example of bipedalism against which to compare and contrast the human condition. And how could I, in good conscience, walk away from such a thing, let alone the mystique of the possible existence of an unrecognized primate in our own backyard?
So I have a question from Bill Brock. He's the host of the Rogue Bigfoot show, and this is a question that you, of all people, are going to have to give the best answer for. The question is, do you think the mid-tarsal break gives Sasquatch extreme sprinting capability?
No, it doesn't. I mean, it's not an adaptation designed for sprinting. I mean, humans are sprinters. There was a documentary where they pitted a human against a racehorse in a 50-yard dash, and the human won because we are designed for that. I mean, our long legs, our stable foot that provides a platform and lever with a speed ratio of our heel to forefoot. Those are all things that are for sprinting as well as endurance running. I mean, physiologically, we're adapted to endurance running and walking. But the Sasquatch, I mean, the mid-tarsal break, as it's manifested in a terrestrial biped, should be best seen as an adaptation for negotiating very steep, broken environments where that flexibility allows for the prehensile function of the forefoot, in spite of the loss of the divergent big toe, or the loss of divergence of the big toe, I should say. The toe's still there, just the divergence is lost. But then the heel can then still act independently while the forefoot provides a basis of support and prehension. All apes are capable of bursts of speed. I mean, if you've ever seen chimpanzees displaying or fighting or chasing one another at the zoo, they can motate very rapidly over short distances, but they're not going to run a marathon. They don't have the capacity to do something like that. Chimps also, they, especially those that are actively patrolling their home range, will often cover a certain amount of distance. Not a huge amount, a mile or 2 kilometers, few kilometers, as they patrol the perimeter of their range. But they're not long distance walkers. So I don't know if that answers.
Yeah, it does, because the terrain.
I get so many people who think that the mid-tarsal break, and I guess in this instance the term break is an unfortunate one, because it's viewed as a disabling feature, one that makes the foot less functional and debilitating. I've had this argument with several uninformed podiatrists, for example, who poo it because they say they couldn't walk, and they immediately point to humans with fallen arches and a Lisfranc fracture of the foot are debilitated. Well, we're not talking about a dysfunction. We're not talking about a pathology. We're not talking about a broken foot. We're talking about the normal anatomy and function of a non-human primate foot. and it's very different. And the failure of those individuals to be able to recognize that distinction just, it makes the discussion pointless, quite honestly. But also recognizing the distinctions of a non-human foot morphology.
Team Taser approved!